for this lecture, we are going to look at how things move across the cell membrane. Now, if you recall, last lecture we looked at the cell membrane. We talked about how it's a phospholipid bilayer. And the analogy we use, which is probably a good place to start for today, is the cell membrane is a lot like a house. That house has windows and doors that we can allow certain things to come in if we want, or certain things not to come in. When it comes to cell membrane, some things are going to pass through fairly easily, like gases. Other things, like glucose and other compounds, maybe proteins, won't pass through so easily. And so I think this idea of this doors and windows is a good place to start, because if we're looking at how a cell functions, a couple of the key things need to occur is we need nutrients to get in. This might be glucose to form ATP. This might be lipids to help form other lipids to help be part of the phospholipid bilayer. It might be proteins to allow proteins to come in and have various different functions. Proteins could be in the form of aminopeptides, like different hormones. And we need things to leave the cell. It may be waste from the cell. It may be different things that were formed in the cell, like pancreatic beta cells forming hormones. It might be neurotransmitter from a neuron. And so things have to enter and leave into the cell. And how these are transported is very much going to be affected by what's the size of whatever needs to be transported. What's its shape? Is it lipid soluble? Some things like steroid hormones, steroids or lipids, are lipid soluble, and they actually easily diffuse. We'll talk about that in a minute. The plasma membrane, where other things are not lipid soluble and will not go through. Electrocharge is going to play a role. Some things that are neutrally charged, like some gases, will pass easily through the cell membrane. Other things will not. And so understanding that there are some of these factors, similar to house and doors and windows, that are going to affect how well certain things pass through the cell membrane. So there's basically four, and there's subgroups of these, four processes we're going to look at. This slide sums it up, and then we'll go through each of these in a little more detailed. Diffusion, which is something moving down its concentration gradient. And so the good way to think about this is if you were to pour, I don't know, some balls or some M&Ms out of a packet, they would diffuse across an area with very little resistance to them, right? Facilitated diffusion, which basically is just diffusion but needs to be helped typically with some protein transporter. Osmosis, which is the movement of water across the plasma membrane, across concentration gradient. And then filtration. And like I said, we're going to get into each of these a little more in-depthly here in a second. All right, so let's look at diffusion. Again, diffusion is very easy to picture because it's something we experience in life, to be quite honest, where we are going to see things diffuse, move away. This might be a stain on a shirt. Not a great example, but it can diffuse from an area where it's highly concentrated to an area where it's slowly concentrated. This occurs across the cell membrane as well, where we're going to have things diffuse from a high concentration to a low concentration. The best example of this probably are gases, where we have, when it comes to oxygen going into a red blood cell, it diffuses from an area of high concentration to an area of low concentration, and oxygen enters a red blood cell. By the way, CO2 works the exact same way. It's going to go also across the same concentration gradient. Gases are not the only example, but it's a good example that we see with red blood cells. It happens every second. That's diffusion, very, very common, moving down this concentration gradient. Another subset of that, in a way, is facilitated diffusion. And this is where we have a concentration gradient, but there's something about whatever this substance is, and their example here is glucose. And glucose is a good example because there is a concentration gradient of glucose. There is more glucose on the outside of a cell membrane, on the inside of the cell membrane. However, glucose is too large. And so it needs a transporter to move it across this concentration gradient. If this were a mucosal cell, There's actually a name for this transporter. It's Siglet, a glucose transporter. On cell membranes, we have GLUT transporters that will facilitate the movement of this glucose from high to low concentration. And again, these are proteins. We talked about this in last class, that these proteins make up large parts of the cell membrane, and they're going to allow, in this case, our glucose in, but not allow anything else in or out for that matter. So facilitated diffusion, again, fairly common way to get things in, potentially into the cell. Now, another term which I know we all heard in high school, perhaps you were like me, and I was sitting in social studies class in high school, fond memories, and fell asleep with my head on the book, and this teacher catches you and says, "You're not going to learn by osmosis that way." And, that wasn't really true because there's no water involved. It wasn't like I was sweating. But the idea that there is movement of something, in this case, is across the cell membrane. Now, what makes osmosis unique is that we're talking about the movement of water. And we're talking about the movement of water across a semi-permeable membrane. That's going to allow movement of water, but maybe not necessarily solute. Now, a good way to maybe think about this, and we see this in a lot of places in the body, the digestive tract, the small and large intestine, is a place we see this a lot, where there might be a high concentration of solute, say in the large intestine. And if it stays there for a lengthy period of time, it will literally draw water across the semi-permeable membrane, in this case, the intestinal wall. That wouldn't be bad. That wouldn't be a good thing necessarily long term. But that would be a movement of water because of the concentration of solute. And so we call this osmosis. And this movement of water can be incredibly important. This can cause movement of water in and out of cells. It can cause cells, if they get too much water, to get too large. If they are too small, they can lyse. And so this idea of osmosis is also another way in which we move something, but in this case we're moving water. We're going to talk later on in the semester about water. Water is going to play a role in electrolytes. We'll talk about that when it comes to the kidneys. But this movement of water is a key process, another key thing that we need to move across the cell membrane. Okay. Let's skipped ahead there. And so we have some terms here when it comes to osmosis. And the example your book uses is a red blood cell, though it's not the only example, where we often say that there is a condition that exists dependent upon how much solute concentration there is versus water concentration. And so we could say that in a normal situation, we have what's called an isotonic. Iso meaning same. The idea that at equilibrium maybe is a good word, where we have equal solute on the inside and on the outside of the cell membrane, and therefore the concentration of water is similar, and we have what's called isotonic. Okay. Well, let's say you have a situation where we'll call it hypotonic, where you have a lower solute concentration that's now going to cause water to now move in to the cell. It's going to move across the cell membrane where there's higher solute concentration. And because there's higher solute concentration inside the cell, water follows along that gradient, and the cell will enlarge. The solution will become hypotonic. The cell, in this case, the red blood cell, gets swollen. The opposite would be if there's too much solute on the outside of the cell membrane. We now have water leaving down this concentration gradient, and now the cell will shrink. Crenated is the term we use. And both of these conditions can obviously be issues. Red blood cells, as far as their movement through blood vessels. Here, if they shrink, just simply their ability to function. And so this idea of hypotonic, hypertonic are going to be two key terms you want to understand as far as osmosis goes. So all these processes we've talked about so far are dealing with transporting whether it's water, whether it's gases, whether it's compounds, molecules, down some concentration gradient without the need for energy. However, there are processes that do require energy. There are times we need to move something against its concentration gradient. Sodium potassium pumps are great examples of these. Cells have these to keep a membrane potential. Remember we talked about last class, a negative membrane potential. Well, we have pumps that do that. Well, those pumps require energy. And so active transport is a very common one. There's a couple different types of active transport we'll see, as well as endo and exocytosis, which we'll get to in a second. So let's look at active transport. Active transport we're going to see a lot in numerous different mechanisms in the body. And so the best way to describe this is the movement of something, some molecule, against its concentration gradient, and therefore requires energy for this protein to carry whatever that is. All right. This requires energy, ATP. We've talked about ATP previously. And typically these pumps, these channels, we'll call these pumps. Ion pump is a very common one where it'll require some ions. Sodium, potassium pumps are very common ones. Calcium, particularly in skeletal muscle, magnesium in other types of cells. These pumps are going to play a role in utilizing ATP to get in whatever it is we need to get in. Exchange pumps are when we see multiple ions being exchanged. Sodium, potassium pumps are a good example of those. We're going to see these a lot. As I said, particularly when we talk about how neurons depolarize, how muscle cells work. We're going to see this in nephrons as well, in the kidney, and how things are transported across those different tubules in the kidney. So active transport. But we don't want to ignore these last two because endocytosis and exocytosis do happen, particularly in larger molecules where they really can't fit across this phospholipid bilayer very easily. And so what we'll often see in endocytosis is a molecule binding to a protein on this layer. The cell membrane begins to actually change its shape, forms what we call vesicle, and then this will then enter in to the cell. So there's still protein receptors playing a role. The membrane, the phospholipid bilayer we're still getting through there, but this is clearly a different way of doing that. We're not diffusing through the membrane. There's not movement of water in this case. Typically we're talking about a compound. And this is kind of a good example here. Again, this is straight from your book as far as receptors being involved in this. Receptors are typically going to be involved in endocytosing these. And our last example is going to be exocytosis. Somewhat similar. We're not involving protein receptors here, but if we want something to leave through the cell membrane that might be too big, it might be multiple molecules, multiple compounds. We see this with hormones actually. Cells in adrenal glands that are producing hormones need to exocytose, get rid of these hormones so they can get into the blood and onto whatever tissues they need to affect. Exocytosis is a good way to do this. So we have whatever this is, it's stored in a vesicle. This vesicle fuses with the cell membrane and then will release into extracellular fluid. In the case of a hormone, hopefully into the blood. In the case of maybe neurotransmitters, it would be into a synaptic space, things like that. And so each of these we are going to again see when we talk about other systems in the body. The next chapter we're going to look at is skeletal muscle. And when we talk about skeletal muscle, how muscle cells contract, well, that's all initiated by a neuron. And neurons, they're called motor neurons, will almost connect to muscle cells. Well, not surprisingly, we are going to have changes in the plasma membrane in neurons. We're going to have neurotransmitter leaving the neurons, being exocytosed. We're going to have on the surface of muscle cells protein receptors that are going to depolarize, that are going to have receptors for various different hormones and proteins. So we're going to revisit a lot of these terms again when we talk about muscle function.